Okay, so hello everyone, and welcome to the third edition of the Open BioML Journal Club. Today we are joined by Andreas Bender, who is a professor of molecular informatics at the University of Cambridge, for a talk about the present and future of artificial intelligence in drug discovery. Thank you so much again, Andreas, for joining us, and I think I will now let you go ahead.
Sure, sounds good. Actually, it says I just tried to share my slides. Host disabled
Oh,
participants.
yes, I'm sorry. All right, you should be able to share the slides now.
I think now it works. Let's see. All right, does that work? Can you see that and hear that? Yeah.
Yeah.
Okay, that sounds good. Yeah, thanks for inviting me to speak here. So, in some sense, it will be a talk maybe different from the previous journal clubs. I don't know, I haven't been there, but this will actually be a presentation mostly about data, data and drug discovery. A few years ago, I started thinking about the data we have and why algorithms in drug discovery are not as successful, apparently, so far, as we would like them to be. And so, one of the things I realized is that the data in our area are actually quite different from many other areas. And that's what I will mostly outline here in this presentation, because it's really important to understand the domain you operate in. So even if you're familiar with algorithms, let's say, right, I mean, the data that you get from AlphaFold, for example, on very different areas, or from customer behavior, for example, right, that looks very different to drug discovery data. And that's what I will outline on the next few slides. So any statements I will make on my capacity as an academic. So we also started some companies before, Helix, Farm-Enable, and so on. But this one is actually an academic talk, so there's no company message in that. It's a review talk, if you wish so. And much of the thoughts here that will be presented are actually outlined in more detail in those two articles here. So artificial intelligence and drug discovery, what is realistic, what are illusions, was an article published in Drug Discovery Today last year, received quite some wide coverage. As a result, I was talking to the German Chancellor, Bill Gates, as well, and so on. So, and those articles actually outline many of the thoughts in more detail. So it's open access, so anyone can read that. I would recommend to do that in case you're interested in how to use data efficiently in drug discovery. So what will I go through today? A few slides about the current state of AI in drug discovery. Then I will talk about method validation and how can we actually validate a model. That's a key question. If you have something that's cool and new and great, how do you actually show that it's better than what has been done before? So how do you validate the model? How do you benchmark? And that's very difficult when it comes to drug discovery data. I will outline that. One of the reasons why drug discovery or AI in drug discovery hasn't had the impact yet that we would like it to have is the data, as I said, data and proxy assays. So we'll go into more detail there and conclude with a few slides on psychology that certainly has some impact as well in the field, the hype cycle and where we are now, and the translational gap of methods. And I will conclude with a slide on, well, so how do we move on from here actually? So where are we right now? So we are in what I would call this third wave of computers and drug discovery. There was one in the 80s, one around 2000, one is maybe today, so every 20 years or so that appears to happen. So that's a fortune cover from 1981. That was the year when Silicon Graphics was founded. So that was really the O2 or Octane. I still remember some of those machines myself. So these were really machines that helped computer graphics greatly. Probably the graphics here was rendered with those machines as well. So that's a fortune cover here proclaiming the next industrial revolution, designing drugs by computer at Merck. So that was very much driven by structure and graphics and visualization. Then there was one wave around 2000. That was also the year when the human genome sequence in the draft version was published. And that was also the year, of course, of the biotech bubble, for example, right? Bioinformatics became a household name. So that was the wave that was driven much more by data. So the first wave really graphics, visualization, structure. The second wave around 2000 was driven by data. And that's also what happens today, more or less, again, 20 years later. So these are some of the headlines that we see today, how AI is changing drug discovery and so on. And that actually implies something, right? And that was 2018, 4 years ago. It implies that artificial intelligence is actually changing drug discovery, right? But according to the outcomes that we see when it comes to approved drugs, for example, I doubt that that's the case. I will present some data on that on a slide. So maybe that perception on those headlines is a bit distorted, in my opinion. And also the headlines look actually similar, right, between 81 and around 2018. So what's actually similar? What's different? What's different is certainly we have more data. We have GPUs, TPUs, deep learning, very powerful algorithms, right? So certain things are certainly different. But what is similar, in my opinion, is in many cases we don't know which data gives us a certain signal. Yeah. And that leads us to doing quite a lot of hypothesis-free research. We sequence everything, and then the question is, well, where can we find a signal? So we do some GWAS analysis or so, right? So needle in the haystack problem. And quite often we don't really know which data to generate. Biology exists in many, layers, but they're not static, right? So you have sequence and expression, protein. You can phosphorylate your proteins, you can activate them, and so on, right? It's a dynamic system, yeah. Every cell is different as well. And so we don't really know how to characterize biology. And that is the fundamental problem then, which data do we actually need to predict something? Yeah? And if you think about biomarkers, for example, that's a tricky topic as well. Yeah, you hardly have biomarkers that have high recall and precision at the same time, for example. Yeah? So basically, it's very noisy. You deal with a very messy system in biology, and that's still a fundamental problem. And we didn't really address that, so we don't know in many cases which data gives us a signal for efficacy and safety in the clinic. If you want to do drug discovery, right, it's not about deep learning, it's not about docking, it's not about data. Yeah, it's about having a compound that's safe and efficacious in the clinic. So not in some model system, in the clinic, right? And we quite often don't know which data actually tells us that a compound is safe and efficacious in the clinic. That's a very fundamental problem. Yeah, it's not a problem of more data or more layers in your network. It's actually a problem of what's the data that gives us a signal. Yeah, and if we don't understand that well enough, that problem will stay with us forever. Yeah, so that's a fundamental problem. That didn't really change, in my opinion, over the last decades. 2000. So that was the second era of computers in drug discovery, right? I'm still old enough to remember that. So in many cases, you have a technology, right, and you have some type of hype cycle, right? And so around 2000, the human genome sequence was published. So this is what 1 year later was written in a review article. 2001, the number of drug targets will increase by at least 1 order of magnitude. Target validation will become a high throughput process. There will be 3,000 to 10,000 targets compared with 483 at the time. Yeah, so a huge number. But actually 15 or 16 years later, there was a Drug Discovery Today article, and that put that number of drug targets at around 667. So not 3,000 or 5,000 or 10,000, right? It's just 50% more, right? So what does it mean? Quite often, you don't actually get what you wish you would be getting when you surf on that hype cycle. On the other hand, the Human Genome Project, of course, was essential. Yeah, we can catalog genes. Yeah, I still remember the times when we didn't know receptor subtypes as well. Yeah, so we didn't even know how many genes we have. There were ideas we have 100,000 genes or so as humans. Yeah, it sounds ridiculous now, but it's 23,000 or so. But those estimates were around. Yeah, and so in this sense, we can catalog biology. That's great. But the question is then, how does it translate to discovery? Yeah, and this is where there's still a weak spot. Yeah. So the question is, how do we go from technology to applications? And what are the limitations of what we do that we need to keep in mind? So what do we see in the data we generate? Yeah. Does sequence tell us enough? Well, quite apparently not as much as we hope to, right? And that's probably the case for AI and drug discovery right now as well. So there's lots of focus, and that's great. We need to analyze our data, but we need to have the right data to draw the right conclusions. Yeah? And I will give some examples in the next few minutes. So there's quite a lot of funding going into AI and drug discovery. It's maybe 20 billion or so in the last 10 years. That's a plot. It's probably very approximate data. But there's quite a lot of focus and hence quite a lot of expectation as well when it comes to the area, right? So that comes from venture capital firms as well as big pharma companies, of course, that heavily invest in their AI departments. So I was working at Astra, for example. So there's really probably is a 3-digit number of people, so in the hundreds of people that work in central AI groups, right? And then you have AI groups in the different disciplines as well. And that's still ongoing as well. So there's some VC funding going to small companies as well as into big pharma, right? So there's quite a lot of expectation in the area. We will come back to that number when it comes to putting things into practice. And this is input, right? There's always input. And then the question is, what do we get as output from that, right? But that's the input that goes into the AI and drug discovery area. And what do companies actually work on? So here on the left-hand side, there's a plot of clinical and preclinical programs in what they call AI native companies. So that's 20 or so companies that put AI first. So Exscientia, Benevolent, and there's a quite large number of companies in the area. They looked at the top 20 or so. They're actually summarized in this plot. And on the right-hand side, part B, that's the big pharma companies. And switch on the pointer. Yeah, so on the left-hand side, that's the AI native companies, and that's the top 20 pharma companies. And in green, that's discovery programs. And then the further you go up in the graph, that's phase 1, phase 2, phase 3 programs. Yeah. And what you see is on the left-hand side, lots of green. So basically, in AI native companies, you have virtually all 95% or so of projects are in discovery or preclinical, right? So there's hardly anything that moved into phase 1 to the clinic. And if it moved into phase 1, hardly anything of that subset again moved into phase 2. Yeah? And phase 1, that's safety, and phase 2 and 3 is efficacy, right? In different sizes of cohorts. And basically, what does that plot tell us? It tells us that in AI native companies, there are hardly any compounds that actually made it to the clinic and that were shown to be safe and efficacious, right? And that's the proof point of AI in drug discovery, of course. Yeah. And if you go back to the headline a few slides before, right, how AI is changing drug discovery. Well, I mean, according to the compounds that went into the clinic, have shown safety and efficacy. Actually, there was not that much change, right? And on the right-hand side, big pharma, that looks quite different. There's actually the number of discovery preclinical programs going down, probably more disease focused. Maybe there are other reasons. But you have many more assets that went to clinical phases, right? So the projects are much more advanced, right? So that looks quite different. And so the point here is we don't really know whether AI in drug discovery works, right? Because there's no marketed drug that was discovered using AI. There's actually, if you go back to some of the old Vertex compounds, the first HIV protease inhibitor was actually using structure-based drug design, right? So today you would probably also call that AI. That's been decades ago, 25 years ago or something like that. And that was using docking, right? So maybe that's a proof, right? But that was docking, structure-based drug design. So here, the current wave of AI native companies in drug discovery have hardly any compounds that went into clinical phases, or none that went on the market, right? So the proof point is still outstanding. That's the point. So what do companies look at? So here you see on the left-hand side, I will only comment on part A here. You see which target classes do AI companies work on in the top 20 pharma. And you see there's much more focus on kinases, on non-kinase enzymes, and there's less focus, or virtually no focus, on nuclear hormone receptors, transporters, and transcription factors. So I thought about that. What could be the reason? There are two reasons, I think. There's lots of data for kinases, enzymes, also GPCRs, right? These are target classes in the 80s, GPCRs, in the 90s, kinases more or less, and then epigenetics targets afterwards. So these are target classes that companies worked on quite a lot. So basically there's lots of data, and those target classes have simpler pharmacology. If you have an enzyme, you can inhibit that and you're done in many cases. It can be more complex, but if you have an ion channel, yeah, or if you have nuclear hormone receptor and so on, then you actually have much more complex pharmacology. You have different subunits. Yeah, the nuclear hormone receptor can bind in different ways and so on. So you can have different transcriptional changes as a result. So the pharmacology of those target classes is much more complex. Okay. So what do AI companies actually focus on? On areas with lots of data and that have relatively simpler pharmacology. Yeah, so you're more likely to be successful, but you're also less innovative, right? That's the trade-off here. So unfortunately, it doesn't really go out of the domain into novel target areas. Yeah, so the biology that those companies work on is actually quite conservative, more conservative than you might think. And the same is true on the chemistry level. So here are some programs that are displayed, and you see in gray discontinued drugs, in a red non-AI derived, so I mean man-made, so to speak, compounds, and in green AI-derived drugs. And so those plots, they're UMAP plots. So you plot high-dimensional space into 2 dimensions. Can be quite misleading, but I was looking at the chemistry as well. The conclusion is true, I think. It's actually the chemistry is not very novel either. Yeah. So you might have seen some of the papers where there were actually only simple bioisosteric replacements that were done. So where simply one functional group was exchanged by another. It's standard practice in medicinal chemistry, but it's not really novel. Yeah. And so unfortunately, both in the biological domain and the chemical domain, there's less novelty present than you would expect. Yeah. So there's no real out of domain or whatever you want to call that, some real de novo work going on. And that's the problem right now, yeah. For novel target classes, you don't have data, right? But you need that for generative models and so on. But right now, there's quite a lot of focus actually on areas where the data is, right? And that is then the trade-off to novelty, yeah. There's less novelty in biological space and chemical space than you would expect, basically. Yeah, so lots of activity, but actually there's little coming into the clinic. Appropriate question to ask, where's the novelty? And data is, of course, limiting. Yeah, and always keep that question in mind. Is input success, right? Lots of funding goes into our output, and there's lots of money going into the area, but there's little in the clinic or beyond, nothing that's approved right now. Yeah? And of course, you need to translate, right? So how do you actually translate all of that input, data, effort, money, and so on into output, right? That's still outstanding, unfortunately. And compare what you expect always to baseline null hypothesis, right? So the first AI designed drug will be celebrated by the media. You can bet on that. But if 20 billion or so of funding went into the area, even your null model would give you 20 or so approved drugs, right? I mean, there's various estimates of what it costs to discover a drug. Depends on the disease area, depends on whether you put in capital costs and so on. But let's say it's $1 billion or so, then even the null model, you would expect that 20 drugs would be discovered by AI in the near future, right? Or with that money, you could discover 20 drugs or so. Yeah, so that tells you that the representation in the media is then a bit distorted as well, right? Because even the null model would give you that number of drugs. Okay, so that was the first part. That's where we are. Lots of activity, yeah, but the translation is still outstanding. And I hope that happens soon as well in the interest of, well, patients, but also, of course, it's great if algorithms work, right? But we're still waiting for that when it comes to clinical success. So second part. So what is validation actually? And I assume all of you are familiar with the word, but if you validate a model, it's different in image recognition, let's say in drug discovery. It's a core question in science, right? You compare to a baseline, yeah? Is my model better than linear regression, for example? It's also a core question for startups, yeah? If you have a platform technology, we did that a few times before, how do you actually convince customers, right? How do you validate your model? And that's generally, it can be difficult to do. In theory, we establish a method, then we use a benchmark, ImageNet or something, and then we know how well the method works by calculating some performance measures on some cross-validation split or something like that. Ideally, of course, you do prospective validation, but I mean, this is usually how you would validate a model if you come from the machine learning side. But in drug discovery, that's difficult. So labels are either mostly only in vitro relevant, I will come to that word in a few slides, or conditional, so they depend on those. We cannot label our data properly is the point here. I will outline that in a bit. Validation, prospective validation is very costly, so you cannot really do that. Yeah. And it's difficult to sample the distribution in chemistry or project space well. So performance depends heavily on the test set. And that's something I realized when, well, training various models, in particular on chemical data, right? And you have different data sets, and they are generally small. They're very biased because you have many analogs in chemical data sets. Yeah, you can basically have a performance measure, but that performance measure, of course, depends on your data set. But you cannot, but it's entirely dependent on the test set that you choose, right? And given that you have so many biases and analogs, and you don't know how well you sample all chemical space, you don't know the underlying distribution. Yeah, basically you get a number that is based on a data set that you cannot characterize. You cannot put it into context of all available chemistry space, right? It's a very biased sample, and you don't even know how biased that is, and that is really a problem when developing models and trying to evaluate that performance. So that's prospective validation, but retrospective validation is numerically equally futile or actually difficult. And the reason is like predicting yesterday's weather. Yeah, and I will have visualization on the next slide. You can do it, and sometimes you even should do it, but you shouldn't expect that your numerical performance translates to future projects. It's more proof by example, so to speak. But you could argue proof by example. I mean, if you have 100 projects and you compare two different models, and one model is right in 60% of the cases, the other one 40% of the cases, then that model you would trust more probably also in the next project. But because you don't know where you are in chemical space in the next case, you don't actually know if that number translates to anything in the future. On the other hand, drug discovery by definition is proof by example. What I mean by that, sometimes you see this drug discovery pipeline as a process, but drug discovery is not engineering. We don't know the underlying biological laws. So basically, drug discovery and the compound on the market, that's the result of 1,000 or so subjective decisions, right? And those decisions, you cannot quantify which decisions were made to compare which decisions were better. You never get that. You don't have controls, right? So in some sense, that's all you need to do. So you cannot have a numerical performance measure that tells you much, but actually proof by example might be actually the best way to validate models. You use them large number of times and you trust the model that works better on average. But the numerical performance is nearly meaningless on the data sets that we use. That's my point here. And the core reason is sampling is impossible, proper sampling. If you come to biology, sequence space, for example, you have transition likelihoods, mutation likelihoods of amino acids, right? You have distributions. You know which amino acids are used, how often in that organism, and so on. In chemical space, you don't have any of that. You have small data sets. They're very biased. Chemists like analogs, right? So whether it's drug discovery or SAR that you explore, you have very biased data sets full of analogs, and you cannot really discriminate true signal from biases, and that is a huge problem. We don't know underlying distributions in the chemical area. We cannot characterize chemical space, and that's a big problem. And this is a visualization here of models of chemical space and which data we can use when we generate a model and when we evaluate a model. Let's close the curtain just 1 second. Okay, so that is chemical space that we, this visualization of chemical space and the data we use to train a model and evaluate a model. So that's usually the training set you use. You have a validation set, something you would like to call an external test set, but in many cases, of course, it comes from the same underlying distribution, so it's not as external as you would like. But then the problem is your next project might be in a completely different area of chemical space. And why is that a problem? Well, because the properties that a molecule has are very conditional. They're conditional on the scaffold. So basically, if you have a change of your side chain, the impact of that change depends on the scaffold that you have. So where you are in chemical space in absolute terms. So it's not just about the distance or difference. If you think about it in machine learning terms, you can also see it in a way that you have a bit vector, so a vector that describes your molecule, and you don't have enough data to actually learn the mutual conditionality of all your descriptors. And that means that you don't know what a change along one variable actually means with respect to the output of the model. That's a different way to see that, but it's actually the same that is visualized here. And that makes it difficult to really anticipate how good is your model in a real world drug discovery situation. So the same is true for time split validation, right? It's quite prominent in the area, but the question is then, well, your next project will still be different, right? So time split validation is actually this proof by example that I mentioned before. There's also a big, very important difference when validating a model. Quite often we talk about model validation, but it's actually process validation. There's a very good paper by Steve Kearsley, was at Google, he's now at Relay. And I visualized that here. Model validation is only one part of what we need to look at. So this is model validation. We have input data, a model, we have a prediction, we can calculate the performance measure. And then we are happy if the correlation coefficient or AUC or whatever we would like to calculate, if that is very high, right? But that's only the look at the model. But there's much more to it. For example, when it comes to drug discovery, right? There's a compound with a project context, and that compound, for example, is dosed at a given dose in men, right? And that compound distributes in a certain way in different organs, and if my model then predicts toxicity or any other property, right, that is actually, that prediction needs to be put into the context of that particular model. Or if we have a model that can have very different purposes, it can be flag toxic compounds, it can be used to remove 20% of the compounds with the highest clearance. It can be used for virtual screening, where we want to discover some actives out of many compounds, and basically you cannot really validate a model if you don't know what it will be used for later. What is the purpose of the model? Should we focus on high recall at the top and so on? So basically, without context, you cannot build a proper model. You cannot validate it in the right way. So you have this input context of the project, and the other part is the follow-up assays. So that's what I realized at Astra. So the question is always, okay, your model predicts something, and drug discovery, what do you do? What do you do next? The model predicts a compound is toxic. Okay, if you don't have an assay, what do you do? Well, you cannot do anything, right? You need to do, need to have a follow-up assay. You will not cancel a project just because a model says something, right? And so in this whole context is very important. And the same is true, and that will be improving drug discovery, right? To put the model into the context. But if we don't look at both in parallel, that will actually not happen, So we need to always consider what is the model, what is the data and the prediction, and how is the model used. Yeah, and that is nearly more important than trying to increase some numbers, performance or so of a model. Yeah, that's abstract. Yeah, but if you use the model, then those left and right connections actually become much more important and are crucial to improve drug discovery, not just model performance. Now, what to watch out for? Proof by example abounds without baseline. Irrelevant endpoints abound. Always keep in mind something like log P or solubility and so on. That's only a physical chemical endpoint. Yeah? In vivo drug discovery is efficacy and safety in the clinic. Yeah? Everything else is just means to an end. Yeah? Validation that matters includes the process and not only the model and the validation. Imagine you have a virtual screening model, so a machine learning model that picks compounds that are bioactive. Yeah, and the model predicts those 10 compounds, and you can only buy a subset of them, which is very common, right? Then you see that the prospective validation is not only based on model performance, but it also the impact of the process is clear, right? You can only buy some of the compounds. Yeah, so model validation is always process validation as well. One mistake is often that success is ascribed to the model, whereas usually you have an interaction between a model and humans, right? And it's easy to say, well, it was the platform, right? But quite often, in my experience, you can go from 1 million compounds to 1,000 or so with an algorithm, and then you need to have a pharmacologist to look at things. Yeah? It's actually really a two-step process. Yeah, it's hardly ever. Yeah, and quite often it's small numbers validation, trivial successes, as I said, and there's no negative control. Okay, so these are areas where I think validation should improve a bit. There's two resources I can recommend to look at. One is a blog post here at drugdiscovery.net/how-to-lie that I wrote. It's not about how to lie, of course. It's about what to watch out for, yeah, in order to present results of a model in a trustworthy way. And the other one is a Nature Reviews Chemistry article that we published together with Nadine, Marvin, and so on. Evaluation guidelines for machine learning tools in the chemical sciences. So that outlines which performance measures do we have, what do we need to watch out for when it comes to chemical data, and so on. There's also kind of checklist in order to hopefully move towards more reproducibility in our area to make code available, and so on. So if you're publishing machine learning models in the life sciences chemical area, I would encourage you to read those papers as well. Yeah, so what's the Achilles heel of AI and drug discovery? That's data and proxy assays. And that is key. That's the most important part of the talk. So this is one analysis we did in this Drug Discovery Today article. Where can you actually improve drug discovery? So what you have here is net savings. So it's basically improvement because you can map both time and success rates in monetary terms, right? So to map that on one axis, it's expressed in dollars here. So the higher, the better. And on the x-axis is the other different phases of drug discovery, from hit discovery to approval. And we model three things. If you are faster, so the time of a phase is reduced by 20%, the failure rate is reduced by 20% in relative terms, or the cost is reduced by 20% of that phase. And you see that lead optimization, for example, if you're cheaper, you save some money, but the real impact comes actually from quality improvements in the clinical phases. Yeah, so that means the black bars, they are highest. So that means better compounds need to go into the clinic, okay? And that is the key message here of this figure. We need to have better compounds in the clinic. Why is that? Failure rates are high, the phases are quite long, and they're quite costly. Yeah, so if you have better compounds in the clinic, so this is where changes really matter. It's not so much in the early stage, but that's actually where lots of the focus is right now. AI and drug discovery, we have lots of bioactivity data, ligand target bioactivity data, so we can build machine learning models in the hit discovery, hit to lead optimization phase. Yeah, but that's actually not where the impact comes from. The impact comes from the clinical phase because it's so long and costly and so on, and failure rates are high, right? Unfortunately, there we have actually quite little data. Yeah, companion diagnostics is one exception. You might call it more from coming from the bioinformatics area. So you actually have genetic markers, right, that tell you to stratify patient groups. Yeah, and that's an excellent example. Clinical cancer, for example, cancer drug discovery. If you have companion diagnostics, your clinical success rates go from 3 to 9%. Yeah, so it's 3 times as high. It's a fantastic success. It's still only 9%, but it's still much higher than it used to be. That's a factor 3 difference. Yeah, that's one of the things that really make a difference in drug discovery. But sometimes you don't have biomarkers. That's a problem, of course. But here the point is we need to have better compounds in the clinical phases. But that's not where we have the data. So here we'll go through data briefly. We have the flat Earth in vitro view. So that's where we believe the labels of our data is more or less in vitro data. And we have the round Earth view, the in vivo view on data. And that's a bit, in my opinion, the elephant in the room that's often forgotten when it comes to AI and drug discovery. Quite often that is not discussed sufficiently, but that's what I would like to do here. And I will outline why algorithms from image and speech recognition often don't translate to drug discovery. Yeah, there's a big difference between the areas. So that's a simple view on the world of AI and drug discovery. It was more or less also my view until 2010 or so. I did a postdoc at Novartis analyzing high content screening data, so high dimensional biological data, and that really helped me understand, have a more well integrated view on chemistry as well as biological readouts. But this is a simple view on the data we have. Molecular structure, bioactivity data, links it to proteins, we have pathway information, and we have links between structure and phenotype. From phenotypic screening, adverse drug reactions, anything like that, but we have a readout on the phenotypic level. And in this worldview, we believe our labels. A compound is toxic. A compound binds a target, or compound treats a disease, and so on. It's a simplified view because whether a compound is toxic depends on dose. Whether a compound binds to a target depends on, in the brain, let's say, whether it actually reaches the brain. Yeah? Does a compound treat a disease? Well, depends on dose, depends on disease endotype, and so on, right? But in this simple worldview, we believe our labels, and that can be helpful. It can be helpful, for example, for mode of action analysis. Yeah? So in this triangle, you can actually predict. You have a link between the compound as well as the phenotype. So you have toxic compounds, non-toxic compounds, for example, and you can use bioactivity data to predict targets. So you can do mode of action analysis. Or you can also design focused libraries. That's the chemistry part here. Or you use it for personalized medicine. It works very well in HIV. It's very cheap to sequence the viral genome. And that's the best example for me, actually, for personalized medicine. So you can predict which drug you actually need to use against which patient to have viral suppression. But let's talk about mode of action briefly here. So this is the visualization in that triangle. So you have links between a structure and a phenotype. And for the compounds that have a certain phenotype, you want to predict based on bioactivity data, the protein, so the class bioactivity label class, basically. And that's where this those simple annotations between chemical structures and targets can help. So it's a very simple concept. You encode your molecule, for example, in circular fingerprints. You can do that very quickly. And then you train a random forest. In many cases, deep learning works a bit better, but trains much longer. You need to worry about your architecture and so on. So with random forest, you probably get 90% or so of that performance as well. And then you can predict targets. So you put in the structure here, for example, here are the top 8 or so targets listed of this staurosporine-like compound. And you can rank by score. So you can have a anticipation of the bioactivity profile just from the structure and based on hundreds of millions of data points you have. What are the problems from the machine learning side? You have many classes, more than 1,000 or so. You have many more inactives than actives, and you have very imbalanced classes. Sometimes you have 20 compounds, sometimes 10,000. Sometimes you have many analogs, sometimes not. Okay, that makes it not trivial from the machine learning side. So you need to adjust your scores afterwards using Venn Aver scaling or something like that. So why does machine learning work here? Because we have lots of data, it's relatively large scale, and it's relatively homogeneous data. Okay, assays are still slightly different. You have different ATP concentrations and kinase assays also, but you have lots of data and it's relatively homogeneous, at least compared to in vivo data. Okay, so this is where machine learning makes sense. Lots of data and homogeneous data. It also makes sense where you have noisy data, but you're only looking for strong signals. So for example, we worked on repurposing using gene expression data. So in this case, you measure an Affymetrix chips, quite a large number of genes and the expression signals. But for repurposing, you only take the top up-regulated and top down-regulated genes. So you don't really model the data. You're looking for extremes in the signal. And that's what you can use to select compounds for, in this case, stem cell differentiation, for example, is one of my favorite papers. It's only cited 5 times or something, but it's still one of my favorite papers because you pick a small molecule, you put it on stem cells, and after 3 weeks, you see a beating cardiomyocyte under the microscope. I find that really fascinating. So using gene expression data and algorithms, we can pick a small molecule to differentiate stem cells and make the cells that we want. I find that really fascinating. Okay, so in this area, machine learning also works. Yeah, we don't have large amount of data, homogeneous data. We have noisy data, tricky data, but we actually look for the extremes, yeah, and look for pattern matching in those extremes between a disease data set and a compound data set. That's also where machine learning works. Yeah, but in many other areas, the data we have is actually much more tricky. Yeah, so lots of text on here. You don't need to read that, but For example, when it comes to pharmacology, the compound doesn't only bind to a target, right? It's an agonist, it's an antagonist, or partial agonist, or something else, or an allosteric modulator, right? You have different downstream signaling, you have different residence times, yeah? And quite often we don't know that, so we cannot label our data properly. Another example, phenotyping. So what is disease phenotypes? When it comes to CNS diseases, depression, anxiety, all those diseases, they are symptom clusters. So you cannot just label your diseases with or compound with the indication of anxiety, another one depression, and so on, because they are symptom clusters, they overlap. And so we cannot label that data either. And that's actually the case for lots of the data that we have in the life sciences. So the key here is the algorithms we have are tremendously powerful, but is our data as well? Well, and quite often we cannot label it. That's the point here. One example, so adverse reactions. Sometimes you see in computer science journals papers on classifiers. Does a drug cause an adverse reaction? Yes or no? Binary classifier. Well, I mean, if you look at that in the real world, then you have a patient with a particular genotype who has a particular disease endotype that you cannot really label, as I said, who takes a particular dose of a certain drug, but sometimes forgets to take it. That's a big problem as well. And that dose of a drug might be taken over a week or over months. So you have repeat dosing. You don't know that in many cases. And then you see adverse reactions only in a certain fraction of cases, sometimes more severe, sometimes less severe, sometimes only if co-administered with a different drug, and then maybe only in males and after 3 months of dosing. So how can you label a drug with an adverse drug reaction, right? You have all those variables to consider. You cannot just say a drug causes a certain adverse reaction, right? You cannot really label your data. The same problem as before, yeah? So does a certain drug cause an adverse reaction? It's certainly not sufficient to have a binary classifier, yeah? At least those you need to take into account, but quite often you don't have that data sufficiently. Here's another example from ketamine. So that's a compound that is actually quite well known, has been used for 50 years or so. It's an anesthetic, it's a street drug, it's an antidepressant, but it's used in different ways if it's dosed differently. IV 2 mg per kg versus 0.5 mg per kg. The mode of action we only explored over years and decades. It's still not quite clear how it works. It seems to work differently in animals where metabolites are active compared to other compared to humans. So the point is, if you put that information then into machine learning models, it's you don't really know, you don't understand even that compound that has been used for 50 years, even that we don't properly understand. So all the annotations we have are on quite shaky ground, and they are very conditional, depend on dose, for example. So that's another example of the difficulty of labeling data. Well, for example, even water can be toxic, right? You wouldn't usually, so if you have a toxicity model, right, how do you label your compounds for machine learning? Well, difficult to do if you cannot just rely on the structure to label a particular compound, right? If you drink too much water, 5 liters or so in a few hours, then you dilute your electrolytes, right? So you have hyponatremia. So that's the reason why even water can be toxic, although you would never label it like that in a machine learning setting, right? So dose really matters, and other conditions as well, of course. Okay, so that's the point here, right? So the data we have, we cannot easily label, which means we cannot train, for example, supervised models so easily. It's very conditional data, and quite often we don't know all the conditions, which were used to derive a particular data point. We don't know the genotype of the patient, where the adverse reaction was observed, and so on and so on. And that makes it very difficult to build machine learning models, to put information into knowledge graphs, and so on. So that's also from this Drug Discovery Today paper I mentioned. There were large successes, right, in chess, Go, and so on, but they are very different from biology. So if you look at the chess Go row here in the middle, so your system is completely determined by the pieces on the chessboard, right? There's nothing external. It doesn't matter who plays. It doesn't matter which day of the year it is or which temperatures it is, and so on. So it's completely, your system's completely defined by the board. It's very different in biology. Sometimes you have stochastic elements, right? You have proteins. Sometimes you have only 1 or 2 proteins that are expressed in a given cell. So you have stochastic processes. You don't know your system basically. You cannot really describe it. So we're poking in the dark using genomics, proteomics, and so on to characterize the system, right? But we don't get all the variables, and that's different from ChessGo compared to biology, for example. And that also has an impact on how we can use neural networks. So in the image domain, CNNs have made great progress, of course, right? But in that area, you have an object here, the cat, for example. You can represent the image in matrix form. you have a convolutional network, edge detectors and so on, that can recognize certain shapes, the eye or whatever, and then you can label that image largely unconditional. Of course, things can be partially hidden and so on, but still you don't have the conditionality present that you have present in drug discovery data. And chemistry is very different. Yeah, this molecule could be reactive, right? Could be protonated in different forms, different crystal forms, and so on and so on. And then the question is, how do you describe that? You could use learned representations, but you still have that conditionality in the data, right? So learned representations assume that you can label your data. If you can't, then learned representations on a few hundred data points that we usually have are really tricky to do in practice as well. So this link from object to representation is more shaky. We don't know how to model. There's no obvious CNN that we can use on a matrix representation. So we need to fiddle around with that. And then we have conditional labels as well. Okay, so you see in the image domain, that is much more integrated. In the chemical domain, there's actually much looser links between those different steps, right? And that makes it much more difficult to translate the successes from the image and speech recognition area with CNNs and RNNs to the chemical domain or biological domain. And one of the main reasons is actually man-made as well. So high throughput screening came up in the 80s or so. So there was the need to profile large numbers of compounds very quickly. And so that's where proxy assays came up. So that don't measure the endpoint of interest, but a simpler endpoint that is easier to measure. Yeah, so one side effect of drugs is, for example, drug-induced liver injury. That's difficult to measure. You need in vivo assays. So there's simpler assays that recapitulate parts of that endpoint. So bile salt export pump, for example, inhibition is one of the mechanisms that can lead to DILI. So that was measured. And so there was a kind of a schizophrenic situation in a way where we are actually interested in vivo endpoints, but what we measure, because that's too cumbersome to measure that, we need to measure that for 10,000 compounds or so. There's lots of data available for those proxy assays right now that try to represent part of the picture of the in vivo situation. Unfortunately, it's only part of the picture. And right now that data, that is what most of the data is that we have from safety profiling assays and so on. And unfortunately, right now that data gets taken into in silico models, machine learning models. And what we have then is a model of a model, right? So we have a computational model of a proxy assay, right? So we're moving further away from the in vivo drug discovery situation. And that's, of course, a problem, right? Because we are less predictive than for the in vivo situation, safety and efficacy in the clinic. And that is then, if we visualize that, that's another reason for this disconnect between model performance and improving drug discovery, because the models are based on those proxy assays. Those proxy assays only resemble part of the in vivo picture, and there's then a disconnect between the model performance, numerical model performance on the assay endpoint, as well as improving drug discovery, which means translating that to in vivo safety and efficacy, right? So that's one of the tricky areas of implementing AI in drug discovery and really having impact in the drug discovery process, right? And it's a detachment of model from process, as well as using proxy assays versus in vivo relevant data. And so this is quite a relevant picture, I think, from the Drug Discovery Today article. There's quite a lot of attention currently on methods and representations, right? So deep networks and so on, but there's all at the top of this pyramid, right? The question is then, we have a method that acts on a representation of data, and that data needs to follow a question or hypothesis. And quite often, and that pyramid should be read from the bottom to the top, right? So we have a question or hypothesis, so we can anticipate DILI using measuring upregulation of a particular pathway, and then the data we generate would be gene expression data. We represent it in some pathway or network representation, and then we use a suitable method, right? That's how you would build it from the bottom to the top. But quite often it's actually quite technology driven. So it's technology push instead of scientific pull. And so that means technology gets cheap, sequencing, let's say. And that means that the data, we actually started the data part, we sequence everything, right? And then we try to actually dig out some information on the data level, but without a proper question or hypothesis in the beginning that gave rise to the generation of the data. Yeah, so it's actually a technology push instead of scientific pull in many areas. But I think to really have predictive models using biological data, we need to start at the bottom and generate the right data for the right question, basically. And only then we need to worry about representation and method. Yeah, that comes at the top of this pyramid. Yeah, so what did AI and drug discovery give us? Patient stratification, that's what I meant before. 3 to 8 or 9%, depends on the area, success in clinical phases. better lookup tables of data. Pharma companies know what they have. That's worth something as well. Some better models, multitask learning, for example, when it comes to related physical chemical properties makes sense. Synthesis prediction can be useful, but actually to then see what the model predicts and match that to the chemistry that can be done in-house can be a tricky part as well. Generative models need some love to stay on track though, otherwise the chemistry they generate can be a bit strange. Better image analysis, certainly. That's something that translates very well. Organ slices, yeah, imaging of organ slices, analyzing that. That actually, I think I would put on the plus side as well. But what it can give us is insight into the translation of early stage data for efficacy and safety related endpoints. Okay, and that's actually key, and that's what I mentioned in the beginning already. Which data do we need to predict safety and efficacy? Yeah, in many areas we still don't know that, and that would be the big breakthrough. So few slides on psychology. As the final part of the talk. So AI is certainly where it is, also because of human psychology, and there's a few factors that contribute to that. So hype brings you money and fame. Realism is always a bit boring, right? There's lots of fear of missing out on different levels, actually. Also on the company level, let's say, right? I mean, if a big pharma company A sees that big pharma company B does something, right? There's some fear that they might get ahead, so there's fear of missing out. Beliefs quite often drive decisions. Maybe someone has the secret sauce, but there's no secret sauce. Secret sauce is doing it, yeah, in my opinion. That's the, it's kind of tongue in cheek a bit, ideal startup strategy equation, yeah. You generate hot air from the startup side, then you bank on fear of missing out from big pharma, and that then creates the perception of secret sauce, so to speak. But of course, you need to validate your platform. Yeah, always see, so look at the numbers, be a good scientist, also in a startup, of course. And there's multiple levels, individual psychology as well as organizational psychology that matter. Yeah? And that's not different from when you were 16 probably, as well as being in a big pharma company. Yeah? There's lots of drivers that go beyond science actually, that lead to decision making in a company. Everyone needs a winner as well. So imagine you're the vice president in a company, you get 20 million to invest into AI, and then a year later or 5 years later, the CEO comes back and asks you, what did you achieve? It's unlikely that you will say nothing worked. Yeah. So everyone needs a winner, right? So everyone reports successes. But if there's then a reporting of successes everywhere, everyone declares victory, and then reality, no one knows what's actually going on, right? And it's difficult to advance a field if there's little real comparison of methods, right? It's very difficult to judge the impact of a technology in this biased environment. And if you put it on the hype cycle right now, where are we? So I only looked at the Gartner hype cycle recently in more detail. So the y-axis is actually expectations, right? There's nothing objective about that. That's important here. It doesn't really exist in this form. It's only a perception. And there's huge spread in the details, depends on the area you operate in. I probably agree with the general place where we are right now. It's probably on top, probably slightly going down already, but certain aspects are clearly working. Deep learning for images, for example, machine learning for target prediction, cloud services, AWS or Google Cloud, are tremendously useful in practice as well. If you want to, in 2 weeks or so, you can have something implemented and running, right? 10 years ago or so, you only would have ordered your machines at that stage, or they would have arrived, right? So you can implement very quickly, yeah. But generally, I would probably agree with that trend here. But still, there's huge spread in the details. Yeah? In the near future, we will then see more about the applicability of a method in a given context. So what survives? Which technologies will get used in pharma over the next 3 or 5 years, right? So there will be some development on that. And so that is the kind of crystal ball, but it's quite clear it's happening already, I think. So capital gets more expensive. So probably 2/3 or so of startups in the field will probably give up. And only those who developed assets, in my opinion, will thrive and develop assets further. But that's less than a third, maybe even much less than a third, right? More data and some successes will be celebrated, but the principal problem we need to address, yeah, which data do we use for which purpose? What do we actually see, safety, efficacy related in the data? And that's unlikely to be addressed. So probably we need to have real consortia involving experimental design. So not just sharing data that is there already, but actually experimental design. So what's the chemical space we need to sample? What's the mode of action space we need to sample? And what's the endpoint space, right? And probably we need really consortia between big pharma companies that involve experimental design to tackle that question, to answer the question, which biological readouts can tell us something about safety and efficacy in the clinic. Yeah, so I think that's the path forward. So, and there's a few avenues that are possible. Omics data, for example, yeah, but we have to see what are the experimental conditions used to generate data. Cell morphology data is another route, maybe organs on a chip, let's see, but it's still under heavy development. Details remain to be seen, standardization, reporting, and so on, possibly organoids as well. Let's see, but that's difficult to do from the practical side, growing organoids in many cases. But there's certain possibilities, right? There's always a sweet spot. So complex enough, but not too complex, right? So you can generate data on a larger scale, but it's still in vivo meaningful, right? Yeah, we probably need those consortia due to the large size of chemical as well as mode of action space that we need to sample. So to summarize, so we need to analyze our data, absolutely. AI is a valuable tool in the toolbox, but the real game changer will only come when we understand biological data better, because we need to bring better compounds into the clinic. It's not about modeling early stage, it's about the clinical phase that we need to tackle. Probably we need consortia for data generation, and methods need to translate into reality. So that's this transition from model validation to process validation that we need to do. Yeah, thank you for listening. Are there any questions? Yeah, feel free. I mean, if you have questions, feel free to just ask them.
I think there might be a question from Moritz.
Sure.
There is a hand raised.
Yes,
Maybe,
you can't hear me, right?
Yeah, I can hear you now. We can.
Okay, yeah, I had to switch my microphone. Okay, thanks. Yeah, well, thank you for this very great and exciting talk and perspective. Like, you were making a very convincing point in the beginning that it's very challenging to develop novel drugs, and one of the reasons you showed was that many new drugs were unsafe as startups failed in phase 1. So I was wondering whether this is really only due to a lack of more meaningful data sets, or maybe also because it's a very hard problem in general to use small molecules as drugs. Maybe the small size of the molecules just makes it too likely to interact with something in the body in an unexpected manner, making them toxic. So while this question might seem very, very naive, and I'm not working with small molecules, so could it maybe be that there's just a relatively limited set of compounds that are safe to be used as drugs, and that we are kind of already converging to exhausting the set in a manner?
So there's two elements here. One is a time element, right? And so one of the reasons is, I mean, drug discovery takes very long, yeah? And you would go, in any case, from preclinical to phase 1, 2, and 3, yeah? Except if you repurpose drugs, because then you jump into a particular phase already. But usually you always go forward, yeah? So it's, in my opinion, it's simply, most of the projects are simply early stage still, yeah? So you would move from preclinical to phase 1, 2, 3. So the point is not that so many failed and nothing survived. Yeah, many projects are probably just still in the first phase. Okay, so don't forget that there's a time element basically as well. Yeah, 30 years later, probably the distribution looks different. Yeah, so there's a time element, but hardly anything did move forward. I don't think we exhausted that space, but there is the hypothesis, the easy cherries or something, the low-hanging fruit has been picked already. Yeah, and there is, some people advocate that hypothesis. To be honest, I don't know, but that is one of the opinions that's out there. Yeah, in my opinion, simply is this translation from the data we have to the clinic. Yeah, that is the problem. Not toxic, lack of efficacy, I would say in many cases. Yeah, so it's actually not that compounds are toxic. I think there's not that much evidence for that, but they are simply less efficacious. They don't work in men. Okay, and I think that is more the problem. Yeah, but don't forget that time element. Efficacy, yes, efficacy will be a problem. Lack of efficacy more. Yeah? Small molecules we have most data for. Yeah, so right now with projects, yeah, or antibody design, yeah, there's so many unknowns. There's 50 years of experience with small molecules. Yeah, so there's lots of data. In principle, it's a good area to work in. Yeah, but the data we have is in many cases preclinical, in vitro data, yeah, and how that translates is then the question. Efficacy, I think efficacy is actually the bigger problem. Yeah? If I kill a cancer cell in the test tube, will I actually kill the cancer in men, which is very heterogeneous? Diffusion doesn't work and so on. Yeah, that's actually efficacious efficacy. I think that is actually the bigger problem, basically.
Great, thanks.
Yeah, feel free, Joseph, if you have a free to do that orally as well. Maybe we have some interaction here. Do you have a microphone? Otherwise, I can read them, of course.
Hi there. Yeah, how you doing?
Good, thanks.
Yeah, so I'll just read the question. You outlined that that AI predicted drugs that we're predicting currently are structurally very similar to existing drugs that we're developing in traditional ways. And I was wondering, and again, this is related to Mortis's question, is do you believe that we're sort of doomed to always predict these similar compounds, given the biased nature that we collect data, where we look at that you outlined, or do you suspect that we can create algorithms and models that do better at generalizing and overcome that?
In principle, yes, but I think imagine you work in a startup company, and you have only 2 or 5 million in the bank or so, and you need to show success. It's more risky to do that. So I think in principle, yes, because you have descriptors that do generalize. What machine learning models are very good at is recombining, and maybe that's actually good enough. If you have kinase inhibitors, you have different fragments that you can recombine, for example. In areas where you have lots of data. In some areas that may be good enough, but it will not be good enough against novel targets, for example, right? That's one of the key limitations. And I think so part of the inability to do that yet is actually based on being reluctant to take the risk. Because if you are in a startup, right, and you have something safer and something more risky, quite often you would tend to do the safer thing. I think in principle, this recombination, recombining different fragments, basically, that could lead to something, in areas where we have lots of data, but it will be tricky against novel target classes, for example. So, but there's more possible than gets tried right now, because of that reluctance to take a risk, I think, in many startups actually, due to understandable reasons, I should say.
Wonderful, thank you so much.
In generalization, okay. Nicole, I wonder though, to what extent, I mean, what did that lead to though so far, when it comes to drug discovery and so on, I would need to, on the chemical level, I would need to be convinced or look at that. So which new compounds were made, how novel were they? And were they actually not only active in the test tube system, again, that would be the in vitro system, did it translate to in vivo, That's the aspects I would probably investigate, to be honest,
If no one has any other questions, I would like to ask a couple, if possible. So one thing I wanted to ask is that I've seen an argument. Well, I generally agree with your argument about the limits of AI in terms of helping out in drug discovery. I've seen one argument though, which I found quite convincing in that when it comes to especially rare diseases that don't necessarily have much funding available for even preclinical work, maybe AI there can have a significant impact simply because it can decrease by quite a lot the cost of the preclinical work itself. And maybe that is sufficient to actually have a pretty big impact on the number of drugs that can be studied at a preclinical level. Do you find this argument reasonable or not?
Yeah. So the important thing is, yes, there's an element to that. And the reason is diseases are related, right? There's an underlying disease context, and quite often in different organs, the same mutation has a different impact. So the disease looks different, but the underlying disease pathway is the same. So that means if in kidney you have a particular disease that's caused by upregulation of one pathway, and it looks different from a disease caused by upregulation of that pathway in the brain or so, you can still use the same drug to treat those diseases. And quite often, rare diseases are driven by the same underlying drivers as well, compared to common diseases. So, for example, to repurpose drugs then for rare diseases using machine learning or bioinformatics approaches, that does make sense. So where you have that underlying link, disease link between common diseases and rare ones, in those areas certainly,
Yeah, thank you. And if possible, another thing I wanted to ask is related to an argument I've seen from David Conarding, who I think is at In-Sitro now, and it's of course related to AlphaFold 2. I think there is a ton of hype around AlphaFold 2, which is unfortunate. At the same time, though, I think the argument itself makes sense, and the argument goes something like protein structure prediction has taken a lot of work by a lot of groups, a lot of pharma groups as well, but there has not been much success until DeepMind came along. And in general, I think it's reasonable to argue that AlphaFold 2 is quite a big improvement upon what previous models. And he essentially asks something like, maybe there is a lot of other problems, maybe not exactly like drug discovery for determining which compounds are actually going to work out in the clinic, but maybe there are lots and lots of problems related or close to drug discovery that can be approached this way. I'm thinking, even you mentioned before data collection, and maybe, for example, machine learning. There has been lots and lots of progress in terms of NLP, and even biomedical NLP in particular. Maybe those can combine, multiply, and have actually a significant impact. Of course, it's going to take time. I think this will be something like medium term, but maybe there is quite a lot of potential, simply because we don't know how many other problems are going to have this phase transition as novel machine learning techniques and modern data engineering methods are applied.
Yeah, so I mean, with AlphaFold, so AlphaFold is excellent for what it does. Yeah, what it does though is structure, and there's nothing wrong with structure, but it's only one small slice. Yeah, so it can help in hit discovery, right? You have the structure, you can docking, for example, you have a hit. The point is though, hit discovery is not really the problem in drug discovery. Yeah, you can do a high throughput screen, also you will find your hits. Yeah, and that way you put the docking for quite a long time as well. So you can, the hit discovery is not really the problem. In AlphaFold, of course, it's a labeled problem, right? So you have over 50 years or whatever crystal structures were generated and resolved, and that's what AlphaFold could use. So you have properly labeled data, relatively homogeneous on a large scale, yeah. And you don't really have that when it comes to in vivo relevant data, yeah. So that's one of the big differences, yeah. For structure, excellent, but structure is only one relatively small part of drug discovery, yeah. If you have a ligand, the question is, how does it translate to the in vivo situation? Yeah? And that's basically not what AlphaFold does, and it's not meant to do. But I mean, that's where the problem is in drug discovery. Yeah. NLP, yes, we have heterogeneous data. That's what we do at Helix, for example, as well. There's quite a lot of heterogeneous data around, and that needs to be abstracted using NLP. Yeah. That is right. That is right. And that data, but the question is always that conditionality in the data and so on. Yeah, that problem still exists. It's the right direction, yeah, but the underlying problems basically when it comes to data conditionality, and they are difficult to resolve using NLP alone because the information is just not in there. No?
Yeah, thank you. Yeah, it makes sense. Thank you so much. All right. I'm not sure if anyone else has any other questions. All right, otherwise we can call it here. Andreas, thank you so much, seriously, for the opportunity and for joining us today. And it was an amazing presentation. So yeah, thank you.
Yeah, great. Thanks a lot. Cheers for the invitation. Huh? Thanks, everyone.
Thank you. Bye-bye.